And may I now invite Professor Sophie to come to the stage to deliver her lecture. Professor Sophie, please.
Thank you, Professor Chen, distinguished guest, Vice Chancellor. It's an honor to be here. It's particularly special to deliver the Shaw Prize lecture at Shaw College, so I'm really honored. And of course, I'm deeply honored to share this prize with Adrian and to receive this wonderful prize recognition of the work we've done over the past 20 plus years. But most importantly, for me, it's particularly special to deliver it at the Chinese University of Hong Kong, given the new partnership between Baylor College of Medicine and the Chinese University in Genetics. I expect a lot more exciting discoveries to come, and we're really honored to have this partnership. So today I'm going to tell you the story of Rett syndrome, how I got to work on it, and how briefly tell you how we got to the gene, but then really move a little bit beyond that and share with you some of the new work we're doing in this area. And I think many of you heard that my first encounter was through a patient. I was still a pediatric resident at the time, and Ashley came to the genetics clinic, and actually she was seen in two clinics at Texas Children's Hospital, the neurology clinic by Dr. Percy, and I was happening to be rotating in genetics, and I saw her with Professor Riccardi, and she was previously a healthy baby up to about 2 years of age, was able to say a few words, sing nursery rhymes with her parents, and sociable, use her hands to feed herself, did everything you would expect an 18-month or a 2-year-old girl to do. But one day, all of that stopped, and she stopped running to the door to greet her father when he came from work, and she became more socially withdrawn. And with time, she lost the ability to use her hands, and I'm going to show you in this video how she was spending most of her time. That's the year I saw her, in October of 1983, when she had these symptoms. She was about 5. You can tell she cannot pick up anything with her hands. You see her walking. She's not quite steady. And you'll see that she's mouthing her hands and have these repetitive stereotyped features. And here she's also hyperventilating a little bit, breathing fast, and then holding her breath. So that was the case of Ashley. And she was referred to us by her pediatrician, who had happened to read the paper, Marlene McAlevey, who happened to read the paper by Professor Hagberg in 1983. He published a paper describing the syndrome and decided to call it Rett syndrome in honor of Andreas Rett, who had actually discovered it in 1966, but wrote about it in the German language, so nobody really knew about it until Professor Hagberg and colleagues published in the Annals of Neurology. And when we read the paper, basically every symptom of Rett syndrome was in Ashley. And you see her here with a little bit of an abnormal gait. So it was, for me, intriguing that the child was normal and she lost all the skills. But it was just one isolated case. And really, I don't know if my fate would have changed if not for a serendipitous event where a week later, I was in my neurology clinic. I picked a patient with a diagnosis of cerebral palsy and decided to see that girl. She was a 12-year-old girl. And she walked into the clinic and had also all the features of Rett syndrome listed on the slide. Now, I recognize that that girl had Rett syndrome the minute she walked through the door because she was 12-year-old and wringing her hands constantly. And as a child neurologist, there was really no other disorders that I have seen that do that. So you see on this slide the features that these girls had. As I mentioned, the language disappear. They look autistic early on, and then they can't use their hands, and they will develop seizures, balance problems, and breathing abnormalities. Having seen 2 in 1 week of a syndrome that neither I nor my attendings have heard of, I gathered there must be more. So I asked the clinic volunteers, I gave them some symptoms. I said, find me girls that have, at the time we called it mental retardation, now we use the term intellectual disabilities, balance problems, seizures, bring me all charts that has all these symptoms. And the clinic volunteers went in search and came back. I reviewed about 30 charts, and in those 30 charts, I found 6 patients that had Rett syndrome. Examined them all, and they all really looked very similar to Ashley. And as a neurologist, there's not much we can do. Once you've done the EEG and once you've done a scan and you really can't find anything helpful, the only thing left to do is a spinal tap. So I did spinal taps on all 6 girls, and we measured the metabolites of dopamine and serotonin and noradrenaline. And lo and behold, we found that the metabolites of dopamine and serotonin were reduced, and we thought that was the only one thing, the only real biological finding that's altered in girls with Rett syndrome. So we decided to publish the paper, and we wrote it and published it in the New England Journal of Medicine in 1985. And once that paper was published, of course, it was the first US series on Rett syndrome to be published. You can imagine many families who had girls that fit the diagnosis recognized it, and now recognize that Houston, maybe there's some expertise, and we started getting a lot more girls with Rett syndrome, and within a very short time, I was able to evaluate and follow about 200 girls with Rett syndrome, and they were all girls. And that's really, I mean, once I've seen those 6 girls and realized how interesting and devastating this disease is at the same time. It's interesting because they're born healthy and normal. They regress, but it's not a degenerative disease. They can live a normal lifespan. Some may die prematurely, but they can live a normal lifespan. That's really what was intriguing about it. And of course, we couldn't do much to help them. And I thought if I go back to the lab and maybe do research on this, I might learn something. And because they were all girls, I thought the best thing to do is to do genetics, that maybe we will identify the gene. And I asked Art Baudet to take me into his lab so that I can learn how to do molecular biology and molecular genetics. And Art was very excited. I told him I have 200 girls with Rett syndrome, and I want to find the gene. And he was really excited, and he said, show me the families. And I did. And those were the families. That's a typical family with Rett syndrome. It's really an isolated case, one in a family. And it didn't take Art but a few minutes to tell me, no, you can be in my lab, but you can't work on Rett syndrome because there's no way you're going to find a gene for a sporadic disease in 1985. PCR wasn't quite discovered yet when I joined his lab, so you can imagine. So he really wisely advised me to pick a Mendelian disorder, and that's why I picked the spinocerebellar ataxia, which I'll be lecturing on Friday. So with that, I sort of kept working on Rett on the side, and when I moved also into my own lab. And really, the working hypothesis was that it must be an X-linked disorder. You can't just search the whole genome. You have to narrow your hypothesis to at least something logical. And because they were all girls, the most logical thing was it's on the X chromosome. And about 4 or 5 years into working on this, I got a couple of families like this one, where you would see that the mother is healthy, but she has 2 daughters with Rett syndrome. And she has 2 different, the fathers are different for these daughters. So to me, that suggested that the gene must have come from the mother because she's the only common parent. The fathers are different. And the only way a mother could be carrying a gene mutation and be healthy is maybe if this is really on the X chromosome. And with the hypothesis being in females, each X is active in about half of the cells. We have, because males have a single X chromosome, for females to be equal to males, they have to inactivate one of their X's in every cell. And this is random. Sometimes it's the maternal X that's inactive, sometimes it's the paternal X. So the hypothesis was that in females with Rett, it's 50-50. But in this carrier mother, somehow she has favorable X inactivation, so it's only the healthy X chromosome is being expressed in all of her cells, and not the one perhaps carrying the mutation, and that could explain why she is normal. And I did the experiment and showed that's indeed the case. So with that, then, use these 2 girls and then 2 other girls from another family to find if they share any region on the X chromosome. And that got us to a big region at the end of the X chromosome in XQ28. and just went really gene by gene, systematically sequencing and characterizing the structure of the gene in order to study the coding part of it. And finally, in 1999, we identified it to be the methylcytosine binding protein 2, the gene that encodes for that. And as you probably are aware, Adrian has discovered this protein in 1992, but at the time, its association with brain disorders was not known. So when we found mutations in it, that was, the connection. And it's really nice that today you're going to hear the story from both of us to hear both sides of the coin. Now, what I'd like to do today with the talk, to tell you what happened after cloning the gene, I'd like to walk you through a little bit what we've learned about the human disorders that could be caused by mutations in this gene, and then tell you a little bit what we've heard about the effect of this protein on brain function, particularly the balance. We have to have a balance in how our brain cells work, so we'll talk a little bit about that. And in the last third of the talk, I'd like to discuss with you some therapeutic interventions we're testing in hope that one day, one or more of these would be helpful for Rett syndrome. So the first thing that we did after finding the gene is, of course, go back to all the DNAs we had in the lab and see, do they have mutations in this gene? And indeed they do. We find that about 95% of girls with Rett syndrome have mutations in this gene. My belief is the other 5% is probably outside the coding region, or we haven't quite found them yet. But what we also learned is that mutations in this gene can cause only partial features of Rett syndrome or very mild cognitive deficits, sometimes cognitive problems with seizures, sometimes behavioral problems such as anxiety and obsessive compulsive disorder, and sometimes just classic autism. I'm going to show you now a video of a girl who actually has classic autism. She was referred for that diagnosis, and she has a very common mutation, T158M, that is seen in classic Rett syndrome. And you're going to see her walking normally. She can walk easily. She's not wringing her hands, but she's not looking at people. She's socially a little bit aloof. She's following command here. My colleague, Dr. Glaze, now will ask her to point to her body part, and she does that quite well. So she can follow command, but she has features of autism, which is lack of social interaction, lack of language, and some behavior problems. So she has favorable X inactivation, favoring the healthy X chromosome. Most of her cells have the healthy one, so therefore she has a milder features. We also learned that in males, there are a few males who have mutations in this gene. Because the males have a single X chromosome, it's very hard for them to survive when the gene is mutated, because they don't have the other 50% normal functioning cells. So most of the males that have an inactivating mutation of the gene, where the gene really loses its function completely, they will die within the first year of life. But there are some males that have milder mutations. And those with the milder mutations typically present with cognitive problems, psychiatric features, sometimes juvenile bipolar, sometimes juvenile schizophrenia, but always with cognitive deficits and tremors. Tremors seems to be a penetrant phenotype. And some may present with autism. And these, particularly those with the psychiatric features, really have very, very mild mutations. I'll share with you the story of one such family. Here's an alanine to valine substitution. This is usually, we consider this a conserved amino acid change, yet it is, when you have it within a key domain of this protein, the methylcytosine binding domain, it is consequential. And I had the chance to examine these two brothers who presented with autism, hyperactivity, aggressive behavior, and one of them had seizures. But then talking to their mother about the family history, her siblings had also the same problem. What's interesting, however, is that the brothers, her three brothers, presented with social behavior and learning issues when they were young, just like her boys. But as they were older, they had now more behavioral problems. When they got to be adult in their 30s, they developed motor problems. And when they were in their 50s, they became wheelchair-bound, and they had autonomic problems, and eventually died prematurely. So this one family teaches us so much about the importance of the function of this protein, and teach us that the social cognitive features are probably the most vulnerable. Because when you have a little bit of loss of function of this protein, as in the case of the alanine to valine, the first thing you're going to see is the autism-like features, intellectual disability. The motor system is probably more resilient, and we see those features in their 30s. But then eventually, even with such a mild mutation, just like the males that I told you, they die if they lack the protein, those males die prematurely, although it takes them much longer. So this is sort of to give you a feel for the broad clinical spectrum we see in patients with these mutations, the loss of function mutations. Now, moving from the humans, we and others, Adrian in particular, have made mouse models of this disorder. And we're very fortunate in the field of Rett syndrome that the mouse actually does reproduce the features of Rett syndrome. So if I was to tell you the phenotype of the female mice, they go through the learning and memory problems, hypoactivity, ataxia, seizures, altered sociability. And the males, just like the male humans, die prematurely. And what's really interesting, in one of our mouse models, which is slightly hypomorphic, so the animals lived longer and showed a lot of phenotypes, we see the same type of stereotype behavior as you see in the girls. Here's a healthy mouse. You can see the forepaws apart, and you'll see with the mouse model of Rett, you'll see these repetitive forepaw movements, very reminiscent of the stereotype activity we've seen in the girls. So we're fortunate that the mouse models here reproduce all the features of the disease. Once we knew that the disease is caused by loss of function of the protein, one of the very early experiments we wanted to do is test the idea that perhaps we can add the gene and maybe replace the function of the protein by adding the gene. And as a prelude to testing gene therapy, we did this genetically by adding the gene using all of its control elements. We used a 90 kilobase of DNA that had the promoter, the 3' untranslated regions, all the parts that controlled the expression of MACP2 in the right time and the right place. So this way, we've added the gene so that it's expressed exactly like it's normally expressed to the mouse, and we wanted to see, are there any consequences to that? Now, this mouse, I'm going to show you a video of, had 2 copies of the gene. One, its normal copy, its endogenous copy, and the other is the human copy we added. So the level of the protein was twice normal. And to our surprise, we found that having twice the normal level of this protein also causes a progressive neurologic syndrome. And you'll see here the mouse falling, having a lot of stereotyped behaviors, and it will also have a seizure. So unfortunately, this mouse also had a progressive disorder. And when we characterized these mice, while early on we found that they learned better, with time they lost that ability. They became hypoactive, also had many of the features we see in the Rett mice. And these mice, by 1 year of age, also died prematurely. About 30 to 40% of them died prematurely. So when we saw that duplication of this gene, at least in mice, resulted in the phenotype, this is a case where the mouse model and the lab work actually sent us back to the clinic. We decided to go back to the clinic and ask, are there patients with a duplication of this gene? Because now we know that doubling it also can cause a neurologic problem. And we actually started searching for such patients, and we found them. And just about the same time, actually, other investigators have reported on that. Professor Means and Professor Vanesh had also cases where there were large duplications in XQ28. They span the gene, but they had additional gene. And the minimal region contained 2 genes, IRAC1 and this gene. However, I think given that in our case it was only MACP2 duplication, we can safely say that most of the features in the human are simply due to the MACP2 duplication. And what we see is that they can have Rett-like syndrome features. They have hypotonia, seizures, and the males die by 20 years of age. Unfortunately, a lot of them die also prematurely. And just as you've met my first Rett syndrome patient, I want you to meet my first duplication patient, who's Brody, who at the time he presented, he really had the diagnosis of autism. You see him here doing this repetitive behavior, going back to his computer in a stereotyped way, and he doesn't have as much social or language, but these were his only problem at the time we saw him, when he was about 10. And unfortunately, with time, he developed all the additional problems of seizures, breathing difficulty, and motor deterioration to the point where he is now wheelchair-bound. So this is the case in males. Doubling this gene in male is quite detrimental. In females, which are usually the mothers of these boys, it's much milder, again, because of exon activation. These mothers have favorable exon activation, so probably very few cells express the duplication allele. But in spite of that, we find very high incidence of anxiety, OCD, and depression among some of these carriers. And occasionally, when the exon activation pattern is random, they will present with a Rett-like phenotype. So then if we were to summarize all the human and the mouse data, the one thing to remember is the brain is very sensitive to the levels of these protein. We have to get it just right at 100%. If we have none of it, it's quite severe, very early lethality in mice and human. If we have 50% of it in every cell, there are many behavioral problems. And if we have twice as much, you get the duplication syndrome. We also find if you have 3 times as much, you even get a more severe syndrome. These babies will die much earlier, and the mice are much more severely affected. So now that we know that doubling it is also bad, we wanted to understand why and how. It could be that you can think if you have extra of this protein, it might titrate its interactors and eventually causing loss of function of the interactors, or it might interfere with the complexes within the protein works in. So we wanted to compare the duplication and the loss of function model at both a cellular, neuronal, and molecular level to ask, do they look similar or do they look different? And I will just show you a couple of summary slides here. This is looking at hippocampal neurons, excitatory hippocampal neurons. And in this case, we're labeling the synapses in these neurons. And these are glutamatergic synapses. And you can see here a wild-type neuron with a nice, healthy number of synapses. But the neurons from a MACP2 null mouse have much fewer synapses, whereas the one with the duplication has more synapses. So at a single cell level, they're really acting the opposite of each other. But to really be sure about the molecular effects, we wanted to go back and look at gene expression, because you will hear from Adrian that this protein binds methylated cytosine, and obviously when it does so, it's probably affecting gene expression. So we compared the gene expression pattern in the brain of both the knockout and the duplication mouse. And what we found is the majority of the genes are inversely regulated. So what goes down in the knockout will go up in the duplication, and what goes up in the knockout will go down in the duplication. So not only they're really at the cellular level opposite of each other, but even at a molecular level, majority of the gene changes that are probably most consequential are also inverse of each other, which really suggested to us that the duplication is probably causing a problem by enhanced function of the protein, too much of the protein function rather than loss of a function, because otherwise it should have looked more like the knockout. Now, I'd like to just take a second here to tell you that these genes, there are about 2,500 genes that are altered, and we've done this in many, many tissues. Majority of them are altered by 10 to 20%. So we're not talking, this is not like a transcription factor that is going to turn genes on and off. It's probably somehow, whatever it's doing in our genome, it's affecting, its loss or gain are affecting slightly the expression of genes in neuron. But there are some genes that are altered by about 30 to 50%. And those are not many, maybe about 50 such genes that are affected by 50%. And I want to show you an example here of such genes. And some of you in the audience who do human genetics might recognize them, because each one of those has been implicated as a causative gene for autism or intellectual disability, either by loss of one allele, haploinsufficiency, or by a duplication. So 50% change in either direction in many of those genes causes a syndrome by itself, and all of these are altered in the rat brain. This will begin to give you a sense about the complexity of the gene expression changes in the rat brain and the challenge we're facing, how we're going to make that brain better in face of so many changes. And that really leads me to the second approach we've taken. Once we face this reality, with so many genes altered, we realize there's not going to be one or two targets that we can come and manipulate and fix this disease. Rather, there's so many changes. And we decided to maybe pursue a strategy that can look at more neuronal systems or neurotransmitter systems, because those you can pharmacologically maybe manipulate. We knew, for example, that the girls, as they got older, they got Parkinsonism, and we knew L-DOPA helped that Parkinsonism. So we knew, for example, that particular neurotransmitter system, we knew affected motor function, and we knew you could manipulate it by using a pharmacologic agent. So we wanted to begin to ask which symptoms could be attributed to which neurotransmitter system, and can we do anything about it? And to do this, in this slide, you'll see all the features of Rett syndrome, which you don't really need to look at them or memorize them. The simplest way I can put it is think of any neurological feature you've ever seen in a human and that you know of, and these girls have it in some form or another, because the whole brain is affected and they almost have features of every neurological disorder. So what we did then in the mice is we deleted this gene from different neuronal types that encode either a subgroup of neurons or a specific neurotransmitter system. And I'm not going to walk you through all of these. I'm just going to give you two examples, the two most dramatic examples that I think we learn more neurobiology from. But I will mention that for most of these, we would get one or two phenotypes. For example, when we deleted the gene from the hypothalamic neurons, we found that the animals had aggressive behavior and ate uncontrollably, and we were able to pinpoint a molecular pathway that regulates feeding behavior that's altered upon loss of this protein. So this is something we would have never known, because the Rett girls are so sick, they have so many symptoms upon symptoms, and they're very hard to get them to eat. But when you now isolate just a few neurons, you don't have all the other symptoms, you get this hyperphagia problem. So the two I'm going to tell you about, the neuronal types, are both excitatory neurons and inhibitory neurons. And these make up the majority of the brain cells. Excitatory neurons make up about 80% of the neurons in the brain, and they project long distance. And they are the main carrier of information in our nervous system. Inhibitory neurons project locally, and what they actually do is modulate the flow of information in the excitatory neurons. So if you can think of excitatory neurons as cars running on a very busy street or highway, think of the inhibitory neurons as the traffic lights that regulate when these cars should cross a certain intersection. So that's the information. So when there is increased inhibition, typically locally, you'll decrease that flow of the information. So to study the role of this protein in these two types of neurons and learn a little bit what might happen, we deleted the gene first from the inhibitory neurons, and I will summarize all the data on this slide. This is work done by Tuan Chow. And what was really surprising is that this is the first neuron that gave us so many symptoms due to the loss of the gene in this type of neurons. In males, you see basically almost all the features that we see in the complete knockout, including the premature lethality. The only thing we did not see was the anxiety or the tremors. So we saw everything else. Now, inhibitory neurons are made of different types: somatostatin, parvalbumin, 5-HT3A, and the majority of them are of the somatostatin and parvalbumin type. So Aya and Kirsten took on this. Aya deleted the gene in the somatostatin subtype, and Kirsten in the parvalbumin. And again, here I will summarize the data just to sort of get the final message I want to share with you. What you'll notice here, as I mentioned to you, the inhibitory neuronal knockout reproduced many features of this syndrome. What was interesting is that the parvalbumin and the somatostatin each reproduce only part of these symptoms. And the seizures and the stereotypy were typical of the somatostatin, and many of the social behavior and learning and memory and motor functions were from the parvalbumin. There were few features that were not reproduced, and we think these may be attributable to the other neurons. Because we did a double knockout of both, we still did not reproduce them, so we think these are the other neurons. So from the inhibitory knockout, we learned a few really important things. What I didn't tell you is what we also found, that the inhibitory neuron had about a 30% reduction in their functionality. The GABAergic signaling was just slightly reduced, about 30%. Imagine that only 30% was enough to give you all these features and premature death. It tells you about the importance of the excitatory-inhibitory balance. And the other thing we learned is this modularity, that different types of neurons have different vulnerabilities. And I think this is something we would have never discovered using many other methods. If we were to use, for example, something that silences the neurons completely, or something that kills the neurons, it's far more severe. What we're doing when we take MACP2 out of a neuronal type, we're partially disabling the neuron. And when you're partially disabling a neuron, you get to know what are the first things that are going to manifest when you partially disable that cell. And that's something that was really interesting. And I think that's what's relevant to the various human neuropsychiatric diseases, because we don't have degeneration of neurons in neuropsychiatric, barring Alzheimer and degenerative diseases, but for most of the psychiatric disorders, it's really partial dysfunction. It's dysfunction rather than total loss of the function. Now, if we look at the excitatory neurons, and this is work of graduate student Shangling Meng, we got, again, many phenotypes, motor abnormalities, and premature death. But now we get the anxiety-like behavior and the tremor. Remember, those were the two features that were missing in the inhibitory knockout. So again, that was really interesting to us, that at a very partial disablement of neurons, you begin to see which ones are mediated which neuropsychiatric features. So if I was to summarize all these studies, I can sum them up in this one slide. And we have actually done both, knockout and rescue. And we get consistent data from both. What we've learned is partial disablement of excitatory neuron uniquely will give the anxiety-like behavior and tremor, whereas we get many social behavior and learning and memory from the inhibitory neurons. There are some behavior that require both: feeding behavior, premature lethality, and motor function. Either way, we got those phenotypes. And this was quite interesting. But also what was important, these are important essential features of the disease. You obviously need both systems to have normal function in this area. So then what we really learned is that moderate reduction in GABA or glutamatergic signaling can cause a multitude of phenotypes, psychiatric phenotype. And the prediction from that is even a more subtle dysfunction of these neurons might give you then maybe isolated psychiatric symptoms and behaviors. From a therapeutic point of view, we learned quite a bit, because initially we were quite excited by the GABAergic story, thinking maybe if we boost the GABAergic system, that will be sufficient to treat Rett syndrome. But I think this gave us pause that really both systems have to be treated together. So this story is now going to either help us guide at least some treatment, given what we know, or we may have something else, alternative pathway for therapy. Getting back to the Rett syndrome and duplication, I mentioned to you that clinically they look the same. They shared many features, but I've also showed you data that really at the cellular and molecular level, they're opposite of each other. So why is the behavior the same when at a molecule or a cell level, they're very opposite? So we propose that maybe the loss and the gain might cause similar behavior due to circuit malfunction. It could be that at the cellular molecular, they're different, but at the end of the day, either direction of dysfunction might give you an abnormal circuit behavior. And the way to evaluate that is to really look at an intact circuit rather than a neuron in isolation. And this is work of a wonderful postdoc in the lab, Wilou, helped by Ryan Ash, who's a computational, helped her with a computation from Stelios Smirnakis' lab. And the circuit we looked at is the CA1 circuit in the hippocampus with all the connectivity intact with CA3 and using slices from these hippocampi from the animal models. And we studied two types of animal models, the loss, which is a null male, as well as a female heterozygous, because that's the true model of Rett syndrome, and then the transgenic mice with the duplication. And now the way we're going to interrogate the CA1 neurons in their intact circuit is by having a transgene that encode GCAMP, which is calmodulin, a calcium-sensitive protein, fused to green fluorescent protein. So anytime the cell fires an action potential, there is a burst of calcium, which due to the interaction with the calmodulin protein that binds calcium, the green fluorescent protein will fluoresce. And in this video, I'm going to just show you an example of that. If you watch these neurons, they're firing, and I hope you can appreciate that they're firing independently. And we actually record from these neurons and prove that with every green fluorescence, there is an actual action potential. So we know this is a good reporter of action potential. Now I'm going to show you an example from a wild-type animal, and in this case, a transgenic or a duplication animal. So here's the wild-type animal, and you can see few cells are firing, not many, and they fire independently. But I want you to look at the duplication mouse, and you'll notice now that the firing is far more synchronous. There is a lot more neurons firing together. And this is not quite good. We don't want many of our neurons firing together because they have to perform different functions. And in order for us to learn, it has to maintain a certain level of asynchrony. We then quantify that. You can see here the wild type, the null, the duplication, and the female Rett. And I hope you can appreciate each of these is a neuron firing. You can see that few fire together, but more so in the null, more so in the transgenic. You can see many here firing together, and a lot also in the Rett girls. What's interesting is that although the Rett brain is mosaic, it actually has a lot of synchrony. So this then told us that there is more correlation of neurons firing together and enhanced synchrony. We wanted to validate this because I told you this was a hippocampal slice. We wanted now to look into the intact animal, record from the hippocampi of awake behaving animals, and from the same cells and ask, do we see the same phenomena? And this we did in the Rett female mice and compared it to the wild-type animals using this tetrode recording done by graduate student Sarah Key, who's jointly supervised by Dion G. and myself. And you'll see here again, we see more synchrony in the Rett mice compared to wild-type animals. And this is not a sign of seizures because we're doing EEG recording at the same time, and their EEGs are very similar to the wild-type animals. And this shows you here the correlation, much higher correlation coefficient in these animals. So now that we've seen, we finally have a phenotype at a circuit level that is shared between the loss and the gain. So now we can interrogate that to ask what's driving that behavior. And this is the circuit now in a cartoon shape. We first test what happens if we perturb the excitation and inhibition by decreasing the function of inhibitory neurons, giving a drug that partially inhibits GABA receptors, very little bit. And you can see here quickly, when we tip the balance for excitation, we see a lot more synchrony. So we know then that at baseline there is some synchrony, but it's far more increased when we tip the excitation inhibition balance. Now in that circuit that I showed you, there's the excitatory neurons, and there's two types of inhibitory neurons, somatostatin and parvalbumin. So we did either a knockout or overexpression in the excitatory cell, and then a knockout in each of those to see which of those neurons may be driving that abnormal circuit behavior. And I will summarize all these knockout and overexpression in one slide to tell you that at baseline, the increased synchrony is driven by excitatory neuron. But anytime you disturb the balance, then it's driven by the inhibitory neurons. So this is really important because in phase perturbations, that might explain why one will go into a seizure, because you have to have a robust function of inhibitory neurons, and in the case of Rett, we don't have that, and that's drive the synchrony. So the very final thing we wanted to do then is understand in that circuit exactly what is driving that. And to do this, we knew of one type of inhibitory neuron that's stimulated by the excitatory neurons, and then itself, it goes back and inhibits the neighboring excitatory cells. So think of two excitatory neurons, one of them stimulating an inhibitory cell. Now that inhibitory cell is back inhibiting the neighboring one. And that's what would hopefully control that synchrony, right? They can't fire together. Well, what we discovered is that system is not working as well. The excitatory input onto these inhibitory neurons is decreased both in the knockout and the transgenic. Again, that was the one cellular phenotype, physiologic, that we were able to get to that showed us what may be driving this finally similar behavior in the two models. So we learned that both the gain and the loss have increased synchrony, and that it is really the excitatory neurons driving this at baseline, but then with any perturbation, the inhibitory neurons are important, and it's due to abnormal excitatory drive onto the feedback inhibitory neurons. So as we were doing these studies and beginning to realize there's all these circuit abnormalities, we've decided to begin to explore maybe circuit manipulations for Rett syndrome. Again, until we can find a way to treat this disorder, we thought maybe if we can neuromodulate the circuit, we might at least change some behaviors. And the first circuit we focused on was the circuit for learning and memory in the hippocampus. And the neuromodulation we wanted to test is deep brain stimulation. Some of you may have heard about deep brain stimulation for Parkinson's disease. It's used now in over 200,000 patients. And it really helps control many of their motor abnormality. It's pioneered by Professor Ben-Abid. And more recently, it is being tested in a variety of neurological problems, including cognition, again, by stimulating a pathway into the hippocampus. So we decided to test if we can change the learning and memory behavior that's altered in the Rett mouse model by modulating the circuit. And for this work, we collaborated with Jean-Rong Tang, who's the head of our neurophysiology, in vivo physiology core, and his capable postdoc Shuang Hao performed the majority of the studies. And the protocol goes like this. When the animals are adults, 8 weeks old, we do the implantation of the electrodes in the fornix to stimulate the fornix that project eventually to affect activities in the hippocampus. We give the animals DBS for 1 hour a day using a paradigm that's used in the clinic on humans, because we hope that this could at least one day have some translation. And then about 3 to 4 weeks later, we evaluate the animals, and these were all different groups of animals. We evaluated for learning and memory, spatial memory, synaptic plasticity, and for neurogenesis. And this just shows you that we stimulate in the fornix, And we record in the dentate gyrus because we expect that the activation of the fornix is going to elicit action potential in the dentate gyrus of the hippocampus. And when we record, we are also ensuring there's no seizure activity, there's no abnormal activity. And the mice are quite happy undergoing these procedures. You can't really tell anything is being done to them. They behave quite normally. I'm just going to show you a couple of pieces of data. I'm going to show you their learning and memory, spatial learning. When we put them in this milky water, they hate water. And if we hide the platform, they can't see the platform, so they're really struggling to get out of the milky water and get onto the platform. And you bring them every day to the same pool, and every day they want to get out as fast as they can. And if they're smart, they start learning from what's on the walls, the spatial cues. They'll learn that the platform must be in this quadrant. By the 8th trial, they dash straight to the platform, so you know they've learned it. But you want to know if they can remember. So the next day, after they successfully found the platform and they're happy they can escape fast, you take it out. And when you take it out, they're frustrated, but they remember where it was, if they have good memory, and they're going to search that quadrant more than any of the other quadrants. So this is how you tell if the mouse can learn and remember. And when we do this with the rat mice, we see that the wild-type mice learn real quickly, but the rat mice take much longer to learn this task. They're slower at it. Of course, if they can see the platform, they do fine. So their motor ability is fine. We test them before their motor impairments. And then if you ask if once you took the platform out, you ask, can they search the right quadrant? And the healthy mice do, but the rat mice really don't. They search all the quadrants the same. And if you check here how many times they cross the right quadrant, here's the wild type and here's the rat mice. So we know the rat mice have deficiency in this area, and now we can ask what happens after deep brain stimulation. And this is what happens after deep brain stimulation. For the wild type mice, they're fine. They perform similarly. But the rat mice now perform remarkably better. They're almost very similar, actually, to the wild type mice. And then if we ask if they can remember and search the correct quadrant, you see the wild-type mice here with DBS or without DBS, they're the same. They cross it equally. But the rat mice only improve after the deep brain stimulation, and now they cross the correct platform more frequently. So that was one behavioral assay. We've done additional behavioral assays on them. Then we ask, how about their plasticity? Plasticity is, again, a sort of synaptic correlate, if you will, of learning and memory. And this is in vivo long-term potentiation. You stimulate the perforant path, and again, you record in the hippocampus, and you expect potentiation after stimulation. And we see that here in the wild-type animal, we see very nice potentiation after we present them with stimuli, but the rat hippocampi don't have that. And in the wild-type animal, it lasts days, but it's clearly different. And when we do deep brain stimulation on the wild-type animal, we potentiate even more. So clearly, deep brain stimulation has an effect whether the animal has rat or not. But what we're most interested in is what happened to the rat animals. And you see here, after deep brain stimulation, their plasticity is far more enhanced. And if we overlay it over the baseline wild type, they're indistinguishable in plasticity from the wild type hippocampi. So this was quite exciting. It told us at least it restores the plasticity of the hippocampal physiologically. And the last thing I'm going to show you is the neurogenesis. One thing we discovered when we looked at neurogenesis in the dentate gyrus is that the rat mice have much fewer new neurons born, but after deep brain stimulation, both the wild type and the rat mice have increased neurogenesis as shown by these quantifications. This is before DBS in the wild type and before DBS in the rat, and both of them improved for both differentiating cells taking a neuronal fate as well as number of proliferating cells. How about the synchrony? Again, that normalized after deep brain stimulation. Here you see in the rat sham mice, they have increased synchrony, but after deep brain stimulation, that synchrony got back to normal. And that pathway I mentioned to you, that there's decreased excitation to these inhibitory neurons, as you can see here in a MACP2 null cell in a rat mouse. Here we were able to record from a wild type cell or a rat MACP2 null. You see the decrease, and after DBS it's normalized, and the amplitude is also normalized after deep brain stimulation. So what we then learned from these studies is that we improved hippocampal learning, memory, we normalized their synaptic plasticity, restored the synchrony, and increased adult neurogenesis. And what's most exciting to me is that it told us that rat brain is responsive to neuromodulation. This is, as I mentioned to you, is a procedure that is in the clinic for a variety of symptoms. We chose here one particular symptom to study, and we find that the rat brain is responsive. So what's next? I think we need to know how often we have to develop it. I told you we treated for 2 weeks, but we tested 3 weeks later. How long does the positive effect last? And what happens if we did this treatment for a year, once every, say, 6 weeks? We need to know that. We also want to begin to evaluate it for motor function, because that's really very disabling in Rett syndrome. And the reason we want to do that is one could do 2 stimulators, one for learning and one for motor activity in one surgical procedure. So we're looking now into that. But of course, one thing we need to remember is irrespective of what we do here and irrespective of what treatment we choose to try on Rett syndrome, we have to couple it with behavioral, cognitive, and physical therapies, because these girls have been for years in a situation where they haven't learned exactly like a normal child will learn. And of course, the timing of the intervention is critical. Now, in the last 5 minutes, I just want to tell you a new story also that we're quite excited about reversing the duplication disorder that I mentioned to you. And this is the work of Hezi Steinberg. In this case, we have too much MACP2. We have twice the normal level of the protein, and we wanted to decrease it back to the normal level. And that's a little bit easier, actually, than replacing the deficient protein, I must say. And for this, we've done a genetic strategy where we normalize it back to one gene and all the symptoms resolved. But what we wanted to do is use something that can be translatable. And for this, we used antisense oligonucleotide, where we deliver them to the ventricle through a pump put into the brain of the mouse under the skin, where the drug goes into the pump. And this was in collaboration with the company Ionis in California. These antisense oligos are small DNA molecules that bind the RNA, and once they bind the RNA, RNase H will degrade the RNA so you don't make the protein. So what we wanted to do is let the mice mature into adults and develop all the features of the syndrome, and then infuse them with the ASO over a 4-week period, measure the protein level, make sure it normalizes, and now evaluate them for neurological recovery. First thing we wanted to know, if we put the ASO in the ventricle, does it go everywhere? And the answer is yes. Here's different brain regions, and this is staining for the ASO, and you find it everywhere: hippocampus, cerebellum, cortex, etc. We also found that we actually normalize the protein level. Here's the protein level in a wild-type animal, the transgenic duplication, and then the transgenic on ASO in 2 different brain regions. So now we asked, did the behavior improve? I'll show you 2 examples of behaviors. Here's their motor activity. Here's healthy mice, the duplication mice receiving ASO. You'll notice that they're hypoactive, their activity gets back to normal, and they're rearing on their hind legs and entry into the center. And these are tracing of a wild-type animal, the transgenic, less active, the transgenic after ASO, normal activity. We looked at their anxiety-like behavior. Anxious mice do not like to be in these open spaces. They will hide in the closed arm. And we measure how often does the mouse goes to the open arms. The less anxious, the more. Here's wild-type animal. They spend maybe 80 seconds. The transgenics are very anxious, but after the ASO, now they venture into the open arms. This is social behavior, how much they interact with another animal. Again, the transgenic interact less, but after ASO, that normalizes. Of course, object interaction is irrelevant for this behavior. It's not social behavior. And the last thing we looked at is seizures. These animals get really bad seizures when they're about 7 months old. So we wanted to wait till they got that old and now infuse them with the ASO. And this is the oldest we've ever tried a reversal experiment. And at this point, all the animals have seizures, and all of them have abnormal EEGs. So here's the wild-type EEG, here's the transgenic EEG, and here's the transgenic EEG with the ASO. The EEG normalized, and the animals stopped having seizure, which was really quite exciting, because seizures is a big deal problem for these individuals. So then what we learned from this reversibility study in the duplication, you're going to hear from Adrian in the rat model, but this is the duplication, is that if we normalize the level of the protein in normal mice, adult mice, either using genetics or in this case ASO therapy, we reverse the behavior, physiology, and I didn't show you the gene expression data. All those altered gene expression changes also normalized. And what this told us that this is a viable potential therapy. Then we tested it on cell lines from the patients with the duplication, and we could titrate the dose to also normalize the level in the patient's cell lines. And of course, this has broader applicability. This is the first time someone tries an ASO on a dosage disease where you have an extra copy. And I think Down syndrome is the classic dosage disorder. So one could select some of the genes like APP that causes early Alzheimer in Down syndrome and consider this. So what do we have to do for this before we can move into human trials? ASOs are being used in human trials for spinal muscular atrophy and other diseases. So at least they've been used in children safely, and they are, they can be translatable. But before we do that, we have to calibrate the kinetics in mice that have 2 copies of the human gene, because our mice had 1 mouse gene and 1 human gene. We only eliminated 1, safely left the other one intact. We want to do it now when there is no mouse gene, just 2 humans, just like the case of the children, so that we can go from 2x to 1x. We don't want to go from 2x to 0.5x. So that's something we've already created these mice, and we have evidence that you can actually accomplish this, and it's still in the work. And of course, then we have to test the distribution in big animals, because the mouse brain is very tiny. Therapy is much easier in a mouse brain than a big primate brain. And we've also, knowing that normalizing the level of the protein reverses the syndrome, we've also embarked on genetic screens to find gene targets that could potentially be inhibited with a pharmaceutical that could regulate the protein level. And hopefully, next time I'm here, I can tell you about that. So in summary, I'd like to share with you a couple of thoughts. This month marks the 50-year anniversary of Andreas Rett describing Rett syndrome in 1966. He reported in September, and his paper is beautiful. Everything we know today about Rett syndrome was in that paper describing 22 patients. And I would say, if not for the scrupulous attention to the clinical details that Andreas Rett and then Beck Haber did, I don't think we would have recognized this as an entity. And recognizing it as an entity was the first critical thing for us, at least for me, to decide who I'm going to do sequencing on. They had to fulfill all these beautiful clinical criteria that were outlined by Andreas Rett. So I think I'd like to, pay tribute to his memory on this particular anniversary. The other thing I'd like to share is that studying Rett syndrome and finding the gene, that it is caused by a gene mutation in MECP2, has really been helpful in that it opened up the idea that a sporadic disorder could be genetic. Before that, everybody thought to study autism, we have to focus on familial cases. People were struggling to find collections of families with autism. But then once we've known that a sporadic disease could be genetic, more and more people ventured into looking for that. And now we know majority of sporadic, at least autism spectrum disorders, maybe not quite idiopathic autism yet, can be also caused by gene defects. There are hundreds of genes to date causing these classes of diseases and intellectual disability. And of course, the idea that the brain is so sensitive to this protein is fascinating. Exactly why, and is this going to apply to many other proteins? I think this is something we should keep in mind. And then, for me, what's also exciting is that manipulating specific part of the circuit might rescue specific features of the disorder. This is ongoing, and the ASO treatment for the duplication is another hopeful strategy. I mentioned a lot of data that's done by many people. I just like to highlight Ruthie Amir is the postdoc with the highest tenacity, who stuck with me sequencing gene after gene till she found the right one. Many others, I mentioned their contributions, and of course, our collaborators, especially Shuang Hao and Zhenrong Tang, and Ben and Zinyu for the DBS team, and others. I'd like to specifically thank the Rett families and the duplication families. They really have been instrumental in our study, trusted us, provided samples, come to the clinics, and all the funders that have funded the work I presented to you today. It's been really a team effort between foundation, Rett syndrome foundations, as well as NIH and Howard Hughes and Keck and Simons. And with that, I'd like to thank you for your attention. And I would like to invite Professor Adrian Bird to come to the podium to tell you the other side of the story that he has to share today. And we will both take questions at the end. Thank you.